Okay, so it's been a long and interesting journey, I hope, for you, but here we are in the last lecture on Chapter 17 on lipids. And just like how we started, we're going to end with a fairly brief lecture. So I don't know if that's good or bad, but that's how we're going to end this chapter on lipids. A number of times throughout chapter 17, I've made reference to cell membranes, so here's our chance to really look and see where lipids fit into this cell membrane and combined with the proteins that we're going to talk about in a couple chapters, we'll get a better view of what the cell membranes that are so important to all of our cells really look like, chemically speaking. So in general, cell membranes have a lipid bilayer that serves a couple of really critical functions. First of all, it separates the cellular contents from the external environment, and this is very important. We have a lot of different things inside our cells that are present in differing amounts or just present only inside, not outside the cells. So it's important for living organisms to be able to organize, and we have to be able to have separation that allows our internal organization to be different from the external environment. Second, we have two layers of glycerophospholipids arranged with their hydrophilic heads at the outer and inner membrane surfaces, and then the hydrophobic tails interact at the center. And this is important because it allows us to maintain that separation, but it also allows for contents within the cell to move out of the cell through the membrane and vice versa, substances from outside to move into the cell based on this multi-layer, this bilayer membrane. Now you probably learned this fluid mosaic model way back in, I don't know, middle school. Maybe they're even teaching it in upper elementary school these days. And then you probably relearned it all again in high school. And then if you've taken college biology, you've probably seen it at least a third or fourth time. But it contains the proteins, carbohydrates, and cholesterol. And it has unsaturated fatty acids to give it some fluid-like properties rather than being totally rigid. It's also got the proteins and carbohydrates on the surface that allow communication with hormones and neurotransmitters. There's these surface features that, first of all, they identify our cells as self as opposed to foreign, and that can be problematic if you're doing things like organ or tissue transplant. It's someone else's organ or tissue and it doesn't have the right proteins and carbohydrates. So there's certain medications you have to take to decrease immune response and all of that, but it has to do with the fact that all of our cells are tagged on those cell membranes with things like these proteins and carbohydrates to allow our cells to communicate with our hormones, our neurotransmitters, and perform their functions correctly and ultimately make us ourselves as opposed to anyone else. And so there we see it. So pretty much all the major macromolecules we'll talk about. You've got those carbohydrate side chains. You've got the lipid bilayer, and we're talking about multiple types of lipids, right? You've got cholesterol present, you've got glycerophospholipids, and you've got the proteins that we haven't talked much about. Pretty much everything aside from, if you look down at the bottom of the screen, there's the nucleus that's going to contain the nucleic acids. That's going to be it. Those are the four major biomolecules, the carbohydrates and lipids that we've talked about, and then the proteins and nucleic acids that we will talk about. So moving things in and out of the cell through that membrane is actually a lot more difficult than it sounds. It's first of all, we want a different internal structure and at least concentration, if not type of substances present than outside. So in order to keep outside different than inside, it requires us to expend some energy. And then if we want to allow for input and output of different things through that cell membrane, we've got to have some processes to help that along. So diffusion, passive transport, just allows molecules, particles to move from a higher to a lower concentration, right? And it may not just be molecules, it might be ions as well. So that's why the generic particles is the safer way to go here. And so that's just going with the flow, and that's one way to allow for the transport of substances through cell membranes, either into the cell from outside or out of the cell from inside. We've also got facilitated transport, which allows protein channels to increase the rate of diffusion. And then finally, active transport, which moves ions against a concentration gradient. So potassium levels inside the cell, for instance, can be different than outside the cell, and we can force that higher concentration gradient to go against the normal diffusion through active transport. All right, here we see a nice little cartoon showing these different types of transport pathways. As we move into biological chemistry, you'll see lots more cartoons because the things are large and complicated. And, as a chemist, I'm much more comfortable with seeing every atom and being able to look at things like amides and, the fatty acid chains in detail. But in biological terms, we're dealing with large, complex things that are much better represented by things like these cartoonish spheres. So here we have the outside and the inside of the cell. We see our polar head group on the outside and our polar head group of the other bilayer member on the inside. And then we have each of those lipid layer members having their nonpolar tails in toward each other where they can interact positively according to London or dispersion forces. So we've got to be able to allow things out. So we have to move aside to allow for that diffusion, the passive transport. We've got to have some regions that can make nice little holes to allow that to happen, or something that has at least a polar and nonpolar part to it, so it can do just like our cell membranes, have interaction with the polar head group, then the nonpolar tail group, then back through the polar head group of the inside layer. We can also have the facilitated transport, which ordinarily results through channel formation by integral membrane proteins. What that means is the protein goes all the way through the bilayer to allow for that facilitated transport. And then finally, we've got a similar protein channel that is used for active transport, but that requires the input of energy to go against the gradient. So we do have to put some energy in the case of active transport to maintain that difference in concentration inside the cell versus outside the cell. So those are our main types of transport pathways in a nice, hopefully easy to understand cartoon. Okay, here we are at the very end of chapter 17. So we'll end with one final learning check, and this time you're asked, what is the transport of particles across a cell membrane from high concentration to low concentration called? Is that A, facilitated transport? Is that B, diffusion, also known as passive transport? Or is that C, active transport? So if you haven't already made up your mind, please stop the video here so you can make your assignment, and then start us back up to check your work. Good luck. Okay, so notice here we have the transport from high concentration to low concentration. That's the way things would ordinarily flow. So this is an example of B, diffusion or passive transport. So there you have it. Hopefully that's the letter that you picked and you understand why, and you would get this right again if something similar were on the homework or the test. If that's not the case, if you're confused or you just guessed and you're not sure what diffusion is or why it was the correct choice, please do reach out to me for some help. Otherwise, we've finished our work on chapter 17 on lipids, and we'll look forward to seeing you again for our discussion of amines and amides.